Okay, welcome back. Now, what we covered last time we talked was about the cells. Okay, we talked about the plasma membrane, we talked about the nucleus, we talked about all the different organelles that are inside the cell. We talked about the shape of the cell and how that's maintained with the balancing of ions and water on the inside and the outside. We talked about that phospholipid bilayer that helps to create the cell's semi-permeable attributes. Okay, we talked about the balance of sodium and potassium as well. How in a resting cell, there's more potassium on the inside and more sodium on the outside. And how cells have functions. And that's what we're getting into now. We're starting to build on that. We're starting to put all these cells together into structures that are called tissues. Okay, once we get a group of cells that have a similar function, they are now known as a tissue. Those tissues will work together for one united job. Okay? That's what we're talking about now. All these different layers of tissues. Okay? So cells have specific jobs. When we're born, we start as a cell. Our mother's egg and our father's sperm join together and immediately turn to its own separate cell. That cell starts to differentiate into a group of cells. Then those cells eventually turn into 3 layers of cells. Those 3 layers of cells then divide into all the different parts of our body. And what happens is inside of each of those cells, inside of our DNA, that long chain of nucleotides that we have in our DNA, one segment will be turned on. And that segment then determines if that cell is going to be an eye cell, a nose cell, a hair, well, not a hair cell, well, yeah, a hair cell, okay, a heart cell, okay? So all of these cells start to differentiate, that's the name of the process, into all these different types of cells which have specific jobs, okay? If we lose some tissues can regenerate, some can't. Okay, and it's good to kind of have a feeling or knowledge of which can and which can't. These are either called labile, stable, or permanent tissues. For example, heart muscle is a permanent tissue. That tissue, if damaged, will not regenerate, will not create new cells. Same thing with our nerve cells. Okay, it could form scar tissue to make the rest of the heart or part of that nerve function okay, but it will not regenerate. If I take a big machete and cut off part of my skin though, okay, we can regenerate part of that skin if it has the right environment to do so. Okay, well, I shouldn't say machete, cut the skin off. If I make a slice, how's that? Okay, that can regenerate. So different tissues have different abilities to regenerate themselves. Okay, so tissues are a group of cells that have a similar job. We study them through a microscope. That studying of those cells within a tissue is called histology. The studying of a cell itself is called cytology. Okay, there's major groupings we're going to talk about. Epithelium. Okay, let me talk to you a little bit about each of these. We're going to go into them in depth. Okay, epithelium has 2 major jobs. It covers and lines. So anywhere that you could touch the surface of the body, including inside the mouth, GI tract, all of the skin, okay, is covered by what's called epithelium. Epithelium. Connective tissue is going to connect or support the body. Okay, connective tissue. Ligaments, tendons, cartilage. Okay. Muscle is going to obviously create movement. Nerves are going to send all the signals. Okay, we're going to send all the signals. I think that most of those are pretty self-explanatory except for the epithelium. So we're going to go through those. If you don't get it, rewatch the tape. Okay, or let me know. I'd be happy to go over it with you. So we're going to talk about these 4 major types of tissues. Okay, here are some examples that I uploaded. Let's zoom in on this for just a second. Okay, nervous tissue is going to include the brain, it's going to include the spinal cord and all the nerves that go out to the body. Okay. Now there are 3 major groupings of muscles. Once again, this is an overview, so don't go crazy taking notes. We're going to go through this. There are different types of muscle. There's skeletal, smooth, and cardiac. Okay? Smooth is going to be in hollow organs, such as the GI tract. I think a really good example of that is going to be, peristalsis. Okay, peristalsis is the projection or the propelling of foodstuffs through the GI tract. That is done so with smooth muscle. Okay, skeletals are skeletal, attached to bones. Cardiac is the heart. Epithelial tissues forms boundaries between environments. Okay, skin surface, for example, lining of GI tract, lining of other hollow organs. Okay, inside of the stomach, inside of the esophagus, inside of the uterus, inside of the bladder. Okay, it's going to be anywhere you can think of that's covering or lining. Connective tissue is going to support, protect, okay? Bones, tendons, fat, blood's actually linked in there, okay? Believe it or not, and I'll tell you why in a little bit, okay? Epithelial tissue, epithelium or lining, is going to line body surface or cavities. Now, you can see at the bottom there, epithelium actually creates the bulk of glands. Now let me talk to you about what epithelium is. Okay, epithelium is cells that are closely packed together. Epithelium is cells that are closely packed together. They tend to be on body surfaces, covering or lining them. But we also have this subgrouping of glands where we have closely packed cells. So if I were to take a bit of your skin and put it underneath a microscope and look at it, there would be on the top surface of the skin, that covering or lining, closely packed cells right next to each other, tons and tons of them, millions of them, okay? If I were to take out a chunk of my liver, whoops, I hit the wrong side, chunk of my liver, and I look at it underneath a slide, that is also going to be closely packed cells, okay? The difference is whether it's covering or lining or it's forming the bulk of that organ. They're both closely packed cells. We're going to talk about glandular epithelium in the future, okay? Next semester during endocrine, we're going to talk about that, glandular epithelium, but just realize they're both closely packed cells. What we're going to focus on now is going to be lining or covering, okay? Lining or covering. So skin inside of the mouth, down the pharynx, the esophagus, the trachea, okay, all covering and lining. On the surface of the eye, covering and lining. The role of epithelium is a multitude, but two of the major things that it does is it provides protection. Okay, the epithelium on the surface of our skin, also known as the epidermis, provides protection. Once again, if I rub my skin, I'm not going to rub down to the underlying musculature, okay? I'm going to have layers of cells that will help to protect. It also provides, as another separate function in different area of the body, absorption or secretion. If you start to think of the lining of the GI tract, the gastrointestinal tract's primary job is to provide protection by a barrier, but its primary job is to provide secretion, or absorption of the nutrients that we eat. Okay, so along the GI tract, the epithelium along the GI tract's primary job is absorption of the nutrients and water from that. It also provides filtration. We talked about the junctions between the cells in the GI tract being tight junctions. Okay, those help to keep substances out of the bloodstream. Okay, because think about it, the GI tract's a long tube that we're putting food in, that we're absorbing foodstuffs in that tube, but we're taking it out of that tube, putting it in the blood. We want to keep foreign agents such as bacteria and viruses out, so it helps to filter that. Okay, excretion, excretion of substances as well will secrete a little bit of mucus onto the surface of the GI tract, onto the surface of the respiratory tract. Epithelium helps with that. Okay, secretions specialized to glands, we'll get into that in the future. Some sensory reception as well. If you think about the epidermis, which is epithelium, okay, that helps to provide a sensation of touch. Characteristics of epithelium, closely packed cells. Anywhere that we look on the surface of the body, these cells will be closely packed. They are very specialized contacts, okay? They're held together by tight junctions as well as desmosomes, that we talked about last time. Okay, they're held together by tight junctions and desmosomes, depending on where you are in the body. So they have specialized contacts between each other. They have polarity. So if we zoom into the whiteboard, I would like to talk to you about this. The easiest way for us to visualize this is if I were to take the surface of my skin, I'm going to take a little chunk of my skin out, and I'm going to transpose it onto the board. Okay? What happens is we've got layers. That may not be big enough. Hold on. Okay? On the top surface of that skin, we have many, many layers of closely packed cells, okay? And inside of these cells are nuclei, okay? As we get to the top of the skin, we lose that, but for now, I'm gonna put nuclei in. Now, if we're talking about this surface of the skin that I'm touching right now, it would be this free surface up here, okay? This surface is not gonna be free. Okay, it's going to be the deeper surface as we go in towards the center of the arm that's attached to a membrane underneath. Okay, there's a little layer of connective tissue. So we're going to have a top free surface, which is the surface you can touch, and then there's going to be a basal surface or a bottom surface to which these cells are connected. Okay, that bottom part is called the basal surface. B-A-S-A-L, basal. The top part is called apical, just like the apex of a pyramid, okay? Apical surface. So the apical surface will be that free edge, okay? The basal surface will be attached to connective tissue and a membrane. Now, if you look at your notes, it says basal lamina and reticular lamina. We're going to put those together, okay? They are going to be the connective tissue and the membrane that's on this basal surface. I'm going to just put it together as the basement membrane for you. So reticular lamina, and basal lamina, you can lump them together as the basement membrane. And this is going to be the membrane and the connective tissue to which the bottom of this epithelium is connected. Okay, you could just lump those right together. So we have the free apical surface and we have that basal surface to which all those cells are connected. Okay. It is avascular. A means without. Vascularity means blood supply. Avascular, but innervated. So that means it has nerves going to it. If you've ever, this is a great way to figure this out. If you've ever given yourself a paper cut, man, does it hurt? You can absolutely feel it because you have nerves in the surface of your skin. Well, towards the bottom, but in the epidermis. But it doesn't bleed, right? Because it's avascular, that top epithelial part. So avascular, but innervated. And it regenerates, obviously. Okay, it regenerates the bottom of the nose. If you rub these off, okay, and the majority of the dust that you find in the house is going to be, or part of the dust, I should say, in the house, is going to be skin cells. The rest of it's going to be the poop from dust mites. Yuck. Okay, but my point is that even though this wears away, you're going to regenerate more of them. Characteristics of epithelium. Here we go. We already did this. Apical is going to be the top part. The basal surface is going to be the bottom part. I'm on the next slide. The layers of the epithelium started from the top. Okay. Apical surface is the top. Basal surface is the bottom. Now, microvilli and cilia we've talked about. Microvilli will be wavy. Okay, if we're looking at a cell, it'll be waves at the top of the cell to increase surface area so we can absorb more substances. We see this in the GI tract, okay? We see these microvilli. They're increasing surface area so we can absorb more of our nutrients. In other places, such as the respiratory tract, we don't have microvilli, we have cilia. And the cilia are more hair-like projections that help to move substances along, okay? So that's at the apical surface, those changes. Basal surface, now once again, if we look at the notes, let me bring you back out, okay? If we look at the notes, and that's hard for you to see, isn't it? Let me zoom in. The apical surface is the top, we talked about that. Basal surface is the bottom. The basal surface is we're just going to consider it the basement membrane. Okay, so you can cross off basal lamina and reticular lamina. That's going to be a membrane and connective tissue to which those epithelial cells will be connected. Okay, so basement membrane. There's apical versus basal surface. Now we're going to start talking about how the epithelium is classified. Okay, hold on just a second. Okay, good. Okay, now we're going to talk about how the cells are classified. I'm going to keep you zoomed in because I'm an awful drawer, and this is much better. I'm awful at drawing. I don't know if you could pick up that. So I'm going to let you visualize this with much prettier pictures than I could ever do. Okay. So epithelium is classified as its layering and its shape. So let's talk about the layering first. There is simple epithelium, as you see at the top. Simple epithelium is one cell layer thick. You could see the cell shape, and you could see the nuclei in there. Okay, there's only one cell high in simple epithelium. Stratified has multiple cells. Okay, you could see the basal surface, which is connected to the bottom. It has multiple cells stacked up to that apical surface. Okay, simple versus stratified. Okay, so simple epithelium is going to be one, stratified is going to be multiple. Now, the second way that we classify is cell shape. There's many varieties in shapes. Now, let me tell you this right now. You're not going to see these shapes perfectly. We're going to talk about columnar and a cuboidal shape. They're not perfect squares, they're not perfect columns, okay? What you're looking for is with squamous cells, and this is the cell at the top, squamous cells don't have a real definitive shape. They tend to be flat, and they tend to look like fried eggs. And you know, if you crack open 3 or 4 3 or 4 eggs, they can look all different shapes and sizes. Okay, so squamous cells tend to have that very nondescript appearance. They still have a round nucleus in the center for the most part, but sometimes it may be a little hard to see. In the center, we have cuboidal. Now, cuboidal tends to have a round nucleus and a square-like presentation. So it may not be a perfect square, but for the most part, it's as high as it is wide, excuse me, with a round nucleus. That tends to be the real telltale sign, okay? Columnar is going to be higher than it is wide, and its nucleus tends to be oval, okay? And that nucleus can be at the bottom or it could be in the center, but it's usually from bottom to center-ish in that oval presentation. Okay, so these are going to be our 3 groupings. So now you put them together. And I could say, now we are looking at a simple squamous. And what that would mean is we're looking at one cell layer thick of those egg-shaped cells. Or now we're looking at a simple cuboidal. So we're looking at one layer of cube-shaped cells. Or we're looking at a simple columnar, which is going to be, one cell layer thick of those columnar shaped cells, okay? What we'll probably mostly look at is a simple squamous, a simple cuboidal, a simple columnar, and then also a stratified squamous. So now if we're looking at a stratified squamous is going to be multiple layers of those egg-shaped cells, okay? So let's look at an example here. This is also in your lecture book. This is a picture of what's called alveoli. This is the small air pockets of the lungs. And this is an area where we want to very easily transpose or transport, I should say, oxygen and carbon dioxide from the air that we're inhaling into the bloodstream. So we want a thin layer of cells. We don't want a lot. We want to be able to send substances or gases across quickly. So this is going to be, if you look at these, a simple layer, one cell layer thick. And if you look at them a little bit closer and say to yourself, do they look like columns? And I know this is hard because you haven't seen them all yet, but this is the way you want to talk to yourself when you're looking at them. Do they look like columns? Do they look like cubes? Or do they look like a messy bunch of egg-shaped cells? Okay, I know that's hard to see, but yes, these are egg-shaped cells in one cell layer thick, and they're lined up next to each other. Okay, sort of like one egg and then the next to each other. And I know this is hard. This is hard because when we're talking about microscopes, you know, we're taking this microscopic world that we've never seen and we're trying to explain it to you. So this is the general introduction, and then we'll really go over this in lab. Okay, so we'll go over this, we'll go over it in lab, and then it's good to come back to this lecture and to listen to it again to clarify things, okay? So simple squamous epithelium, and this will be found in the lungs. Now, if you look at this one, okay, I'm going to zoom you back out here. If you look at this one, we are actually looking at collecting ducts in the kidneys. Now, one other thing that is new since we're looking at microscopes, keep in mind that this is a two-dimensional picture of a three-dimensional object. So you need to be able to sort of reconfigure structures in your brain. So if I go into the other room and I find a big tube, or use my water bottle as an example, okay? So here's my nice tall water bottle. It looks like a tube, okay? But if I take it, and you can only see from this direction forward, and turn it on its end, okay? What it now appears like is a circle. And as we're looking at this slide, we actually have collecting ducts in the kidneys, and they are, oh, that looks better. You can see that better there. They are columns, long tubes. But what you're looking at is them on their side. They've taken that tube, they've put it on its side. So I'm trying to look at the camera and get, okay, there we go. Now it looks like a circle. So it is a tube, but we're looking at it on end and it looks like a circle. That's exactly what we're looking at in here. There are several tube-like structures that you're looking through. And through those actually pass urine. Still considered filtrate at that point, I believe. But anyway, to be urine, what will be urine, okay? We're looking at it on end, we're looking through that circle. Now on the inside of that circle, we actually have epithelium, okay? Covering or lining that tube, the inside of the tube, okay? Because that epithelium is actually going to determine what substances will be put into and out of the urine, okay? So when you look at this slide, each one of these is a tube, okay? And when we look at those cells, I think the real giveaway to this is each one of those nuclei is round. Now, if you think back to the different type of epithelium that I talked to you about, the cells that had the round nuclei were the cuboidal, okay? If I tell you that on the inside of this tube is a layer of epithelium. Inside of each of those tubes is one layer of cuboidal shaped cells lining that tube. That is going to be a simple cuboidal. Okay, it's a simple cuboidal. Now this is tough. Write down any questions you have and come see me. Okay, so this is a simple cuboidal on the inside of these tubes. Okay. Moving on to the next one. So different types of epithelia and what they look like. There's a simple squamous, which we talked about. It's one cell layer thick. It looks like those fried eggs. Simple cuboidal is going to be one cell layer thick of those cuboidal shaped cells with a round nucleus. Simple columnar. Those simple columnar cells are going to be higher like a column than they are wide with that oval shaped nucleus. Now pseudostratified columnar is confusing, okay? Pseudostratified. I'm going to break the word down for you and then we'll look at a picture and then once again we're going to look at all of these in lab, okay? So pseudostratified columnar. They are only one cell layer thick, but they look like they're multiple layers, okay? Pseudostratified. Pseudo means false. These are falsely stratified columnar cells. They're only one cell layer thick, but they do indeed look like there's multiple layers. Okay, so pseudostratified, false appearance of layers. So here we go. We're back in the alveoli. This is simple epithelium. This is our kidney tubules. This is simple cuboidal. Okay. Now this one, if we look at this one, once again, I want you to think about the three-dimensional picture in a two-dimensional presentation. So here we are with my, let me try to get in front of the whiteboard, my tube again, okay? If I take this same tube, and this is actually in the gastrointestinal tract, if I take this same tube and I put it on its side, okay? I put it on its side, and then I cut halfway through it, like this, and I look inside, it's gonna look like a half pipe. Do you know what a half pipe is? Most of you do, some of you don't. Half pipe is what people ski board on, okay, or skateboard on. So if you imagine that I take a half pipe where it ends up looking like a U and I move it towards you, there's going to be epithelium lining that whole half of a tube. Okay, so if I just take a little section out of the middle, there'll be epithelium lining the tube at the top and the bottom. And that's what's happening in this picture. So the tube has been cut. We're looking at, here's the tube. We're looking at the epithelium that's lining the tube, okay? So we've got epithelium up here and we've got epithelium down here. Don't let that throw you. What I want you to understand from this picture, imagine if that's confusing you, just that we're looking at the bottom half. This would be what type of epithelium? If I'm talking about from here to here, this would be one layer or multiple layers. This would be one layer, if we're looking from here to here, of very tall cells that are narrow, and of course this nucleus isn't oval shaped. For the most part, they look oval shaped. Okay, and then we've got a nucleus that's towards the bottom or towards the middle. This is going to be a simple columnar. Okay, this is going to be a simple columnar. So don't get confused because we've got it on the top as well as the bottom. Okay, this will be a simple columnar. This will be in areas of absorption such as the GI tract. Okay, and this actually has microvilli on the top to help with that absorption. Okay. Excuse me. So let's look at another example. This is like my least favorite picture in these presentations. If we look at this and break it down, if I tell you once again that the epithelium will be from here to here, okay? We're looking at cells that are higher than they are wide, okay? So this is going to be a columnar. Okay, once again, this is not my favorite picture in the world, but bear with me, we'll look at it again in lab, okay? So this is going to be a columnar cell. Now, this is what I don't like. This is hard to see. If you look at this picture in your book, you can see the nuclei are at different heights. Now, this is not a stratified columnar. I'll tell you right now, I will never show you a stratified columnar. It's a very rare epithelium. So if you see an epithelium that looks like it's columnar and all the nuclei are at different heights, it's going to be pseudo or falsely layered stratified columnar. Okay? This is a pseudostratified ciliated columnar. This is what we found in our respiratory tract. Okay? So here it is, here to here. It's higher than it is wide, just like a column with different heights of nuclei. This is going to be pseudostratified. Okay? Now, stratified epithelium is 2 or more cell layers. It's highly regenerative. It's usually in the areas of wear and tear and friction. It's durable. Excuse me, it's got those desmosomes and some tight junctions, which hold it together just like a quilt that's been sewn together. Okay, it gives it durability. And it's usually in areas of protection. Stratified epithelium, skin, mouth, esophagus. Okay, so stratified epithelium. Some examples. What we are going to look at in this class will be stratified squamous. Okay, we're not going to look at a stratified cuboidal or columnar. We'll talk briefly about transitional when we get into KUB. Okay, so stratified squamous epithelium is going to be found on all the surfaces exposed to the external environment. Okay, so here we go. This is a great picture. If we look at this picture and I were to tell you that the epithelium is between my hands, is there going to be one layer or multiple layers? There's multiple layers of cells. And the way that you could tell that is each one of these purple dots is a nucleus. So you could see there are tens to 20s of these cells stacked on top of each other. So this will be a stratified. And if you look at the shape of those cells, they're very mishmashy. They're not real definitive in their shape. They're not that cube shape. They're not that columnar shape. This is going to be a stratified squamous. This is, if you take any samples of the skin, the inside of the mouth, okay, you're going to see this, stratified squamous. Okay, this is transitional epithelium. We'll find this on the inside of the bladder and the inside of the ureters. It's stratified, this transitional epithelium, and it has real bubbly, it looks like soap bubbles all over. Okay, on my presentation. Okay. Glandular epithelium. So once again, we're going to talk about glands in the future. Glands could be one-celled or multiple cells. They could be, we're going to talk about endocrine, I should say, in the future. We're going to talk about exocrine now, okay? They could be endocrine or exocrine. Let me explain that. I'm sorry, I'm getting ahead of myself. So glands, they could be one cell, they could be multiple cells. We've got exocrine and we've got endocrine. Okay, now, let me see, I do go into it. Okay, so unicellular would be one cell, cellular would be multiple. Okay. So endocrine are ductless, okay? They're ductless. They secrete hormones which will travel through the bloodstream to affect target organs. This is a whole chapter unto itself that we'll get into in the future, okay? So endocrine, ductless, they release hormones to the blood which will travel to target organs. Excuse me. Exocrine are going to have a duct. They will secrete their substances to a surface. Sweat glands, tear ducts, okay, are both examples. And then there's a whole list there, sorry. Okay, so it travels by a duct to a surface. There's unicellular and multicellular. So the unicellular exocrine gland that we will talk about is called a goblet cell. This goblet cell is going to secrete mucus to a surface. We're going to see this in the respiratory tract and the GI tract. Okay, I'll show you an example in just a bit. Okay. A duct can be branched or tubed. It can be merocrine or holocrine. Okay, holocrine is going to explode, merocrine is going to secrete just substances from the cells. Okay, so connective tissue. Let me see. I am going to start connective tissue in the next class because it'll tie in together. Okay, if you have any questions about epithelium, absolutely come see me. I'd be happy to go over it with you. Thank you so much, and I will talk to you soon.